The title of this work is Evidence for Tissue Dielectric Property Differences Between Neonates and Adults, a retrospective study using MR-EPT. MRI of neonates has a number of safety issues associated with it. One hazard is that the radio frequency waves used by MRI scanners cause some heating by depositing energy into the tissue. So essentially this means that when determining the safety of MRI scanners, and specifically the safety of the radio frequency coils, we need to consider the dielectric properties of tissue. While adult dielectric properties are well understood, less is known for neonates, as their tissues have a higher water content, which is dependent upon age, and this is known to affect their dielectric properties. Measurements of neonatal tissue dielectric properties usually require taking tissue samples, which is not possible for healthy neonates. Electrical properties can be extrapolated from adults using scaling factors which are taken from animal studies. However, our recent work has shown that if we account for these differences, this can lead to significant differences in the predicted specific absorption rate. There are many existing methods for measuring dielectric properties, and these typically require specific measurements to be made, some of which are slow and prone to motion artifacts. To overcome this, we have used a method called SAEP, because it allows the electrical properties to be estimated from a single scan, solely based on received coil sensitivities. The data was collected as part of the Developing Human Connectome project since 2015, and this means we can make use of the large dataset of archived raw data from neonate scans. For our study, we used data from 627 subjects, ranging from a gestational age of 26 to 45 weeks. All data was acquired on a 3 Tesla Philips Achiever scanner at 128 MHz, using a custom-made 32-channel neonatal head coil, which you can see on the image on the right. The SAEP method required us to use fully sampled images from each receiver coil with a separate reference. This was obtained from the Sense reference scan, acquired from the 32-channel receiver, plus body coil reference, which were also archived for all subjects. To aid with the masking of the brain, we used T2-weighted fast-spin echo imaging with post-process image segmentation. We made an automated pipeline to process the data. The inputs were the complex reference data, as well as a segmented T2-weighted scan used for masking. We then applied some smoothing to reduce the noise, and applied the SAEP reconstruction to determine the conductivity and permittivity. At the top, you can see maps of the conductivity and permittivity calculated for one of the datasets. It is important to note that the bright rim around the edges is caused by smoothing. In order to exclude these bright edges, as well as any regions of the skull which were present, we applied masking, and this allowed us to calculate the dielectric properties of brain tissue alone. The figure on the top shows the reconstructed conductivity and permittivity for an adult, neonate, and phantom. The measurements are noisy, and so large regions of interest were used for averaging. Even though the data are noisy, it is possible to extract meaningful average conductivity values for all the datasets and average permittivity for the phantom. At the top you can see scatter plots for the gestational age at scan against both conductivity and permittivity, where the points are coloured by their gestational age at birth and are sized by the standard deviation for each measurement. The conductivity has evidence of a weak negative correlation against gestational age, with an R-squared value of 0.32. As opposed to this, the permittivity results were a lot noisier and contained many unphysical negative values. For the 416 term-born babies, meaning that they had a gestational age between 37 and 45 weeks, the group average of the mean brain conductivities was 0.968, with a standard deviation of 0.08 Siemens per meter, and the group average median value was 0.985, with a standard deviation of 0.071 Siemens per meter. To put our results into context, at a frequency of 128 MHz, the expected average adult brain conductivity is 0.47 Siemens per meter, and a 0.9% saline solution at 20 degrees Celsius is expected to have a conductivity of 1.4 Siemens per meter. These are consistent with our measurements. Applying scaling arguments based on water content, an average conductivity of 0.79 Siemens per meter is estimated in the brain of neonates, whereas our data found median brain conductivity in term-born neonates to be higher at 0.985 Siemens per meter. There is also some evidence of a decrease of conductivity with gestational age in the neonatal cohort, but this is less certain from our data. Thank you very much for listening. Please get in touch with us if you have any questions.